All right, we can go ahead and get started. So it's my great pleasure to introduce today's seminar speaker, Dr. Graham Strub. Dr. Strub is an associate professor at the University of Arkansas for Medical Sciences. He is a surgeon scientist with dual doctoral training in medicine and biochemistry. His research is focused on uncovering the molecular mechanisms driving vascular and lymphatic malformations, with a particular emphasis on microRNA regulation and on the development of targeted therapies. He leads multiple NIH-funded programs, research programs, and has been recognized with numerous awards for both his scientific contributions and his dedication to mentorship. Today, he will be sharing his work on next-generation treatments for lymphatic malformations. Please join me in welcoming Dr. Strub.
Well, thank you all very much. I'm going to keep the talk relatively informal today, but the goal that I have is to just share with everybody, what we've been doing here at Arkansas Children's with regards to lymphatic malformation research, and sort of to tell the story about how we arrived there and what we're planning to do in the future. I'm going to turn my camera off so you're not looking at me reading a screen, if that's all right with everybody. And then we'll get started. So, hang on one second. Okay, so I was asked to do just a quick introduction of who I am and how I arrived here. So basically, when I was in college, I was interested in biology and chemistry, and that's what I studied at the time. The Human Genome Project was the big thing. And so everybody was interested in that, and that got my interest as well. So I entered a master's degree in genetics. And not wanting to go into the workforce with a master's degree in genetics and wanting to extend my training for as long as humanly possible, I entered an MD PhD program, which was 8 years. And the PhD portion of that was focused in biochemistry and molecular biology. And I started by studying sphingosine 1-phosphate with Dr. Spiegel in Virginia. And that's how I learned, basic molecular biology and about things like microRNAs. And then I went on to go to the University of Washington to do a residency in ear, nose, and throat surgery. I really became interested in ENT when I was a medical student. And I knew that it would be challenging potentially to continue to use my basic science training if I was going to be a surgeon. But I was fortunate enough to go to a residency program that had a lot of emphasis on research. There was an entire year of no clinical work where you just did research, and that was protected by a T32 grant. And so I got to spend a whole year, and I actually was able to do basic science. And I found two mentors, Jonathan Perkins and Mark Majeski. Jonathan Perkins is an otolaryngologist who also does research in vascular anomalies, and Mark Majeski is a vascular biologist there. And it was through that work that I realized that I would be able to potentially work with children, both clinically and in the research realm. And so that's how I got really motivated. I ended up going to ISFA when I was a resident, and after that, it was pretty much decided for me that I was going to dedicate my research to studying vascular anomalies, because there was so much that we did not know, and that's where I wanted to focus my interest. So I came to the University of Arkansas and Arkansas Children's Hospital in 2018 as a clinician scientist. And that means that they provided me some protected research time. It was actually 5 years of protected research time. So we submitted our first extramural grant in 2023, and it was funded. And so I'm going to share with you some of the data that went into generating that grant, as well as some of the data that we've generated since then. But I want to start with just sort of what my roles are. I have the clinical role, so I do a lot of cochlear implants. I see patients with vascular anomalies, and then I have a busy general ear, nose, and throat practice. And then the rest of my time is spent in the laboratory. We have a standalone lab, and we also have a multidisciplinary vascular anomalies team that sees patients, and I'm the research director of that. And then I also do some basic clinical research. Okay, so in ear, nose, and throat surgery, we encounter a bunch of diseases that are difficult to cure. We like, as surgeons, to be able to operate on diseases once and have them stay away and never come back. But there are several diseases in children that do not behave that way, that become very burdensome. And I've just illustrated a few examples here From top left, that you're looking at the airway, that triangle there. That's a stenosis where you have abnormal scar tissue forming that narrows the airway. Then there's papillomas that can form on the airway. These are polyps on the right of that in the nose. These all prevent normal breathing. The bottom left picture is called a cholesteatoma. That's abnormal skin cells proliferating behind the eardrum that eventually rupture out of the eardrum and dissolve all the neighboring tissues that causes all kinds of problems. You may have seen people with these types of scars on the ear. That's called a keloid. These can form anywhere. They're essentially hypertrophic scar tissue that will just continue to grow and not stop. And then we have vascular anomalies, of course. And these also behave in a similar way, where you have this aberrant growth, despite not being a malignant lesion. And so, what do all these things have in common? Well, if you look at the cells that compromise all of these diseases, they all proliferate abnormally. They all trigger some sort of local inflammation or immune cell dysfunction. And they all cause some sort of remodeling of adjacent structures, meaning that you've got this abnormal lesion and the tissue that it's growing right next to also starts to undergo changes, even though it could be a completely different cell type or tissue. And so the point of that is that these are all diseases of molecular biology. So in order to cure them, we need to focus on having tools in molecular biology to address the underlying issue. And really, the underlying issue for me when I think about it as a clinician is what I like to call this benign disease conundrum. So what do I mean by benign disease conundrum? These are just examples of me operating on different patients with vascular anomalies, either capillary malformation on the left with a laser, or a big lymphatic malformation in the neck, or an AVM of the ear, or a venous malformation of the airway, etc. So we have a lot of different ways to treat these, right? We can operate on them. We have sclerotherapy, we have laser, we have some molecular therapies. But the bottom line is, if this was a cancer, right? If this was like a rhabdomyosarcoma, then you would have to take the whole thing out, and you'd have to take out everything that it touches, and anything that's nearby, you would need a margin of tissue. Because if you don't do that, then the child dies of rhabdomyosarcoma either months or years later from metastasis. But in vascular anomalies, we don't have that problem, right? These are not malignancies. So if you've got a venous malformation or a lymphatic malformation that wraps itself around a nerve that controls your swallowing or your facial movements or your tongue function, you can't take that out, right? You can't paralyze a child's vocal cords to take out a benign lesion. So this creates the conundrum whereby we are constantly leaving behind tissue that is abnormal in these vascular anomalies, despite any method of treatment that we use, even if we're meticulous when we do surgery. And therein lies the issue of this disease recurrence and then frequent treatments, different modalities, etc. So the actual technical part of the operation would be easier if it were a cancer, but it would be devastating in terms of its outcomes. So we need better tools to try to address that recidivistic disease after our current treatments. And that's what our laboratory focuses on. So we all know this. These are somatic mutations. These are mosaics. So in ENT surgery, because most of these lesions occur in the head and neck, we see a lot of these. And obviously the disease severity and type depends on, during embryonic development, what cell gets a mutation, when it gets the mutation, and that's going to determine, the distribution and type of each one of these diseases. Okay, so I wanted to just give sort of what I think of as the current approach in modern medicine to these types of diseases of molecular biology. And so, for example, in our situation, we get a DNA mutation, right? So one nucleotide is changed, and what this leads to is the production of some sort of abnormal messenger RNA. That could be too much RNA or not enough RNA or an abnormal form of RNA. And what that does is then lead to the production of an abnormal secondary messenger. And this is a protein, obviously, and this can function in a variety of different ways that cause disease, but primarily they focus on these particular cellular phenotypes: proliferation, survival, motility, invasion, and resistance to apoptosis. We all learned this in basic molecular biology and biochemistry. The point is that it's these cellular behaviors that cause these symptoms, the symptoms that the patients come in and present with, right? Pain, disfigurement, can't breathe, can't swallow, can't see, can't hear, constant infection, hemorrhage, poor wound healing. In the case of cancer, metastases, right? It's those cellular phenotypes that cause these. And the reason I bring up this type of symptomology is because when we think about treating these kids and adults, we have a lot of treatments that have been around for a long time that target these symptoms, right? These are your painkillers, your antibiotics, your steroids, anti-inflammatories, et cetera. And then as time and technology and research improved, we developed treatments for the actual cellular phenotypes themselves. You can think of these as like your traditional chemotherapeutic agents. And then more time goes by and we learn more about molecular biology and we start developing treatments that actually target these secondary messengers. So you can think of your monoclonal antibody types of treatments, or alpelisib is a good example. It works in this way. But if you notice, as time has gone on, we've kind of worked our way upstream on this pathway. And what that leaves to me is that I feel like there's a big opportunity zone in this region where we don't really have any types of approaches to target these functions. And one of the mechanisms that regulates this type of function are microRNAs. And that's where our laboratory is focused on, and how microRNAs function in vascular anomalies, and can we use that information to develop new types of treatments? So just for some background, in terms of microRNA, I always give a little bit of background on these because some people know a ton about it and some people don't know much. You may have heard of them from the recent Nobel Prize in 2024 in medicine for the discovery of microRNAs. These are short RNA molecules that essentially regulate the expression of different genes. And the way they work, I use a construction metaphor to kind of demonstrate this. And so if this is a little too simplistic, please forgive me. But let's say you want to build a building. This is a rendering of our new hospital that we're building here in Arkansas. And you want to build a hospital and you want it to work and you want it to last and you want it to function without problems. Well, if you're going to do that, you need all different kinds of workers, right? You need plumbers and painters and electricians, et cetera. And you can think of those as like different cell types in an organism, okay? So in this metaphor, every single worker, no matter what your job is, gets a master copy of all the instructions that every worker would possibly have to make the entire hospital run. And you can think of that as DNA, right? Every cell gets a copy. But every cell doesn't need every instruction, right? So the plumber doesn't need to know what the painter is doing and vice versa. So you can think of like highlighted chapters or like a Cliff Note version of this gigantic document being handed out to each different cell type and saying, okay, this is your specific job. And you can think of that as the RNA, right? So the DNA is the master copy, RNA is the actual specific instructions for each cell. And then, of course, the cells do what they do. You get your building blocks, et cetera. So in this analogy, your concrete is made up of amino acids, et cetera, and the cells do what they do, and then you build a finished product. Okay, so there's the dogma of molecular biology as told through a construction metaphor. So what is a microRNA, and how does it fit into this metaphor? Well, let's say you're the paver and you're out there paving, and then it's going to rain, and you can't pave in the rain. So the foreman gets on the microphone and says, all you pavers, no paving today until I say, go back out and pave. That instruction from the foreman, you could think of that as a microRNA. It's a temporary instruction from the cell that says, ignore for a moment what you're being told to do by the RNA until this instruction goes away and then resume what your normal job was. That's how microRNAs function. And so in that way, they're able to temporarily regulate and fine-tune the expression of genes. And so what does that look like in a cell? So in all cells, microRNAs are made. So inside the nucleus, they're transcribed just like any other gene. So you transcribe a section of DNA that codes the microRNA. This forms a hairpin, and that hairpin is exported from the nucleus and then is processed by a variety of different proteins to form a short RNA sequence. This RNA sequence matches through complementarity a sequence on a messenger RNA that's being translated. And when it binds, it silences that expression. So you can imagine that we can co-opt this system potentially. You can design a complementary sequence to a microRNA and give it to a cell, and it will essentially act like a sponge and absorb those microRNAs, and then that gene gets expressed. Conversely, you can give a copy of a microRNA and you can actually silence the translation of a gene at will. So that's in vitro. And so the question then becomes, can we co-opt this microRNA system in humans, in human disease? And how do we do that? And is that possible? So in order to do that, what do you need to know? Well, in any given disease, you need to know which genes there are that are aberrantly expressed. You got to know which ones of those genes are pathologic. So those two things are not the same. You may find a lot of aberrantly expressed genes, but they might not mean anything. So which ones are actually causing the problem? And then once you figure that out, you need to know which microRNAs regulate those particular genes and which ones will actually reverse the phenotype. And if you're able to do all of those other things, then you're left with this other really difficult question. And that is, how do you get these microRNA sequence into tissues in an actual patient? And so those are the questions that you're faced with when you're trying to decide, can we use microRNAs to treat human disease? And that's what we've been trying to tackle. So the context that we're tackling this in is in lymphatic malformations. As we know, this is the most common vascular malformation in the head and the neck. And we have a variety of treatments for these that you're all obviously familiar with. You can leave them alone. You can take them out with surgery. You can inject them with a variety of different things. You can blast them with a laser. Usually we're doing a combination of all of those things for complex disease. But the bottom line is that it's difficult to cure these kids. Some of them are simple, like a very simple macrocystic lymphatic malformation you can get out, but that tongue on the top right is going to be a challenge for that patient's entire life. And so the goal obviously is to figure out better ways and additional tools that we can add to this armamentarium of treating lymphatic malformations. All right, so for just some background, I know you guys all know this stuff, but in lymphatic malformations, the gene of interest is this gene PIK3CA. And we know that this gene has a few hotspots. And when you mutate those hotspots, that essentially causes the formation of an abnormal PI3K. And what that does is overall leads to an increased amount of activation of this very crucial signaling pathway. And that increased activation leads to, at least in cancer, the formation of tumors. But in the case of vascular anomalies, we have the same type of cellular behavior, right? We get increased proliferation, migration, resistance to apoptosis, et cetera. And in the bottom right, you can see if you just take normal lymphatic endothelial cells, that's HD-LECs, and you compare them to LMECs, or lymphatic malformation endothelial cells, and you grow them in a dish, and you scratch across it, and you let them grow, you will see the LMECs move faster. If you do proliferation assays, they grow faster. So it fits the pathway. Okay. So we've developed drugs to treat these, right? This mutation's been around for a while in cancer. We've known about it. So there are medications that you can give that systemically inhibit this pathway. The two obviously ones that are most common are sirolimus and alpelisib. But of course, this is systemic therapy. You're knocking this pathway down in all cells in the body. And so it comes with a variety of very severe side effects and difficulty tolerating the medication. You don't know how long you have to give it. And some patients just don't respond to it for reasons that we don't understand. So there is a need to develop better tools. Here's the problem. This looks very simple. This is what we're really dealing with. Here's PI3K, right? So we don't really know if you mutate that, what are the downstream consequences in all of these other potential targets? And that's what the drug is doing potentially, but that's also what the mutation does. And so we don't really understand all of the downstream effects of having this mutation in the long term. And that's part of the ethos of our laboratory in examining pathways other than the actual PI3K pathway because of this crosstalk between multitude signaling cascades. But where do you start, right? I mean, this is a mess. So how do I figure out where I want to start? So when I got to Arkansas Children's, the first thing we did was RNA-seq. I just felt like that was the easiest thing to do. So we isolated lymphatic malformation endothelial cells from 13 different patients, and we did RNA-seq, and we compared it to normal human dermal lymphatic endothelial cells from 3 different patients. And this was just our initial experiment. And what we found was that we had almost 1,300 messenger RNAs that were differentially expressed, either up or down, significantly, when you compared LMECs to HDLCs. So what do I do with this information now? Am I going to do 1,300 Western blots to figure out which one is up and down, and how do I know which ones are pathologic? I mean, what I would usually do is start getting in PubMed and looking at papers for the top 5 up or down genes. But all that does for me is introduce bias because I know when I'm looking at PubMed, I'm purposefully looking for papers that might fit what I think is going on. And I'm probably ignoring some papers that say the opposite of what I think is going on. And I think that that just leads to wasted time and effort and potentially science that's not really robust. So the good news is at Arkansas Children's, I discovered that we had a very powerful multi-omics core and a whole system of multi-omics research that's being done in our Cancer Institute. And so what is multi-omics? So multi-omics essentially is just the overlaying and integrating of multiple different omics platforms. So you've got, for example, genomics, right? So we all know next generation sequencing, you can identify n equals 1 in terms of what mutation you have. Yet somehow, when you do RNA sequencing, we're getting almost 1,300 downstream RNAs that are now abnormally expressed. But if you overlay that with mass spectrometry for all known proteins, what you find is that only 176 proteins are actually significantly up or down. And so what that tells you is that just because you're finding an RNA molecule, an RNA-seq that's 20-fold up, does not mean that the protein is going to be abnormally expressed. And if that's not happening, then you can pretty much cross off that RNA potentially as being pathologic. And then on top of that, you have this microRNA regulation system. So we did microRNA omics as well, and we identified 110 different microRNAs, again, that were either up or down. And keep in mind that these microRNAs, their function, of course, is to regulate the expression of these proteins at the translation level. So then with some very clever integrative omic software, our team at our core was able to draw essentially a map integrating RNA to protein regulated by microRNA to identify specific pathways, not individual molecules, but functional pathways that were potentially abnormally expressed. And in order to qualify, you had to kind of score, you had to be on all 3 of these omics platforms. And what did that show us? Interestingly, it showed us that the 3 major pathways that were always involved were either pathways that controlled proliferation, inflammation or infection, or extracellular matrix remodeling or paracrine signaling. And if you recall, those were like the 3 phenotypes I described earlier in terms of what these diseases do. The cells proliferate when they shouldn't. There's local inflammation and infection. And I described changing of adjacent structures, but you can think of that as extracellular matrix remodeling because in order for cells to change a nearby tissue or cell, there has to be some degree of signaling and extracellular matrix remodeling in the tumor microenvironment, if you will, to enable that to happen. So what we did was we took all this data and we looked for what I call triads, meaning that I wanted to find molecules that had a dysregulated messenger RNA, so either up or down, with a matching protein. So the protein had to be up and the RNA had to be up. And then they also had to have a microRNA that was known to target it that was regulated in the opposite direction. So in the case of messenger RNA being high and protein being high, that suggests that maybe a microRNA, which normally would silence that expression, could potentially be turned off. And that might be why you're seeing an increase in the messenger RNA. And so that's how we went about selecting the different pathways to investigate in lymphatic malformations using this omic data. So how does that work? So let's look at just a few examples. So this is a slide of lymphatic malformation endothelial cell. And the purposes for this slide, anything that's green is upregulated and anything that's red is downregulated. All right, so one of the microRNAs that came up as being super high in all LMECs was this microRNA 21. MicroRNA 21, we validated its expression, and everything that we got on the sequencing, we had to validate with actual experiments. So the first thing we did was we looked at whole tissue on the left and then isolated endothelial cells. So what do I mean by whole tissue? Each one of these lines with the 2 dots represents a patient, and each normal tissue dot is from a biopsy of an adjacent site to the lesion. And the LM tissue is obviously their LM. And when you do RT-PCR for microRNA 21, just on whole tissue, you get a significantly higher amount of microRNA 21 in the LM tissue compared to adjacent tissue. And then when you isolate the endothelial cells from these tissues, we find the same pattern, right? It's in the endothelial cells and it's higher in the LMECs than in the normal HDLECs. Or sorry, in this case, LECs, because this is from patients. Okay, so what does microRNA-21 do? Well, its four top binding proteins are all tumor suppressors. So microRNA-21 inhibits the translation of these four tumor suppressors. And in doing so, by turning off a tumor suppressor, obviously you're going to have increased proliferation. So we looked at our proteomics data. All four of these were down, and we looked at Western blots, and we validated that all four of these were significantly lower in LMECs. All right. And we also saw, I showed you the slide earlier where we compared the two different cells, they proliferate faster. So what happens if you start manipulating microRNA-21 in lymphatic malformation endothelial cells? Well, if you give it more microRNA-21, you see that they all proliferate faster. So in normal HDLECs and in LMECs, the introduction by transfection of more microRNA-21 into cells causes both of them to significantly increase their proliferation. But what about in LMECs? I'm sorry, what about when you inhibit microRNA-21? Because that's obviously what we want to do, right? It's too high. So what if we inhibit it? So we give the complementary sequence of microRNA-21, which again acts as a sponge. So you're sponging up all the microRNA-21 in cells. And what we see is that in HDLECs, we did get a little bit of a decrease in proliferation, but nothing compared to what we saw in LMECs. They basically cut their proliferative rate by about 60%. And when we looked to try to validate what tumor suppressor this was going through, it looks like it's this protein called PDCD4. And PDCD4 is a tumor suppressor that binds the eukaryotic translation factor 4A, and it essentially decreases proliferation. And what we saw on Western blot was about a 60% increase in expression of PDCD4 in LMECs when you inhibited microRNA-21. But we did not see that in normal HDLECs, just a slight increase, maybe 10%. And so the percentage increase in the expression of PDCD4 in both cell types matches the decrease we see in proliferation in both cell types. So this clued us in that this was the mechanism that was operating here. This is important because microRNA-21 is all over the place when it comes to cancer research and wound healing. People have been developing topical microRNA-21 inhibitors or mimics in skin cancers and wound healing for years, and have shown that if you give more microRNA-21, you get a higher amount of proliferation. This is good for things like wound healing. If you inhibit microRNA-21, you get a decreased growth in things like skin cancers, which obviously is good for inhibiting something that's growing when it shouldn't. And so our question is, can we use this information and try to do this in lymphatic malformations? All right, so otherwise for preliminary data, I mentioned earlier that there were proteins involved in inflammation and immune cell regulation. So here are probably two of the most important. This is ICAM1, which is a transmembrane glycoprotein. It's in the immunoglobulin superfamily, and it regulates cell-to-cell adhesion, especially leukocyte adhesion to endothelial cells. And we know that in lymphatic malformations, we have an increased amount of leukocyte infiltration. And then there's CD44, another transmembrane glycoprotein. This regulates cell-to-cell interactions, adhesion, and migration. And increased amounts of CD44 is seen in a variety of different cancers where you have increased proliferation. Again, these both had microRNAs that regulate their expression that were significantly down on our omics experiments. When we started looking, of course, we validated this with Western blots. We started noticing that even if we looked at LMECs compared to HDLECs, it matched this phenotype. So this experiment is when you take LMECs, you grow them on a dish, you let them adhere, or HDLECs, and then on top of them, you culture them with monocytes, and you let the monocytes adhere to the endothelial cells, and then you wash off any of the monocytes because they won't adhere to the plate. So you wash off whatever's left, and then you count how many cells are there. And LMECs, they stick monocytes much more tightly than HDLECs do. And our hypothesis is that this is regulated by ICAM1, and we have some preliminary data that suggests that this is happening. All right, what else? We identified a variety of proteins and genes that are involved in endothelial junctional integrity. Here's just a few of them. All of these had corresponding microRNAs that were significantly upregulated. So again, these are all proteins that are significantly down in LMECs with microRNAs that control their expression that are significantly up. And in particular, angiopoietin-2, I'll talk a little bit more about because we did some, follow-up investigations into that. We haven't gotten to the other ones yet. But what we found, well, I'll show you that in a second. The other preliminary part of the data was the third phenotype I was discussing, ECM remodeling and paracrine signaling, a variety of different genes. In particular, these two, relin and TPA. So relin is an extracellular matrix glycoprotein. It regulates the formation of the functional collecting lymphatic vessels, but it was discovered in the nervous system. And the reason it's named relin is because when it's turned on, it essentially shuts off this growing process of axonal neurons. So it's like reeling it in, if you will. So it's sort of an anti-invade other structures type of protein. And this protein is way down in lymphatic malformation endothelial cells. And the microRNA that controls it is way up. And then on the opposite end of the spectrum, you've got the protein TPA. You may have heard of TPA in terms of like the clot busting drug, but TPA is a protein that's made actually by endothelial cells. And what it does, it's a serine protease. It converts plasminogen to plasmin, and that function degrades extracellular matrix. So if you're a cell, an endothelial cell that wants to, say, intercalate itself into the muscle of a tongue, you have to dissolve the extracellular matrix of those tongue cells. And TPA, if you turn up your TPA, that may be a mechanism by which that occurs. And then we have a bunch of other data showing that there's some circulating microRNAs in the media of these cells, but we haven't really followed up on that yet because we've been so busy with this other stuff. So let me show you a slide from the angiopoietin-2 study. So if you recall, angiopoietin-2 was very underexpressed in LMECs compared to HDLECs. So what we did was we took in a plate, we grew HDLECs, and then we took LMECs from a patient, and we transfected them with either an empty vector or an angiopoietin-2 overexpressing vector. And we injected them into Matrigel and made these little implants under a nude mice skin, okay? And then we harvested them after 35 days and stained them with either H&E or podoplanin to see what, if any, lymphatic vessels developed. And so going from left to right on the panel B, you'll see a picture of just Matrigel. So that's what the gel looks like. So those little dots, those are not nuclei in that picture. And then if you look at HDLEC4, you'll see a bunch of these little dilated, tiny little dilated vessels. And they stain positive for podoplanin. So those are tiny lymphatic channels that have formed in Matrigel. And this has been demonstrated by others that this can occur. So now if you take lymphatic malformation endothelial cells in the third column there, and just those are the ones with the empty vector transfected, you see these giant dilated lymphatic vessels that form. And those are obviously all podoplanin positive. But interestingly, the angiopoietin-2 overexpressing ones showed a significant reduction in this formation. There's still some vascularity that's higher than HDLEC4, but it's way, way smaller than these huge vessels. So it just shows that this is clearly participating in the formation of these dilated lymphatic channels. And the question is, can we possibly restore the expression of angiopoietin-2 in a way that's translatable, right? Because I can't transfect, a kid's lymphatic malformation with this overexpression vector. This is just demonstration of proof of concept that it works. So we're in the process now of using our system of microRNA nanoparticles, which I'll get to towards the end of the talk, to try to target angiopoietin-2. All right, so here's just a bird's eye view of everything I just showed. And these three different phenotypes, these are our three specific aims for the R01. We said we're going to look at microRNAs that regulate proliferation, ones that regulate junctional integrity, and ones that regulate extracellular rematrix remodeling and paracrine signaling. And we've made some progress on all three of these, and I'll go through a little bit of the data that we've been able to generate, since the grant was awarded. I mentioned earlier, how are we going to get these how are we going to get these microRNAs into tissues? Well, people are already doing this in wound healing and skin cancer. And that's good news for us. I basically just do what the cancer guys do, like about 10 years later in vascular anomaly. So, so these are two panels from papers. One's looking at wound healing, one's looking at skin cancer. But the bottom line is, if you create a nanoparticle with a hydrophobic and a hydrophilic face, and you load it with microRNA, which you can, these will be absorbed not only by cells, but they can cross things like skin. And that's our goal, essentially. Because we're fortunate enough that the majority of these vascular anomalies, at least that we treat, communicate with the skin in a way that you can see them, then it provides us an opportunity to potentially administer a medication, or in this case, nanoparticles, that will not only cross the skin, but can actually get into the lesion itself. Now, obviously, you could inject these as well, but the dream of mine is to develop an ointment or a cream with microRNA containing nanoparticles that will silence these aberrant pathways that you can apply, twice or three times a day, instead of having to take a systemic medication that's inhibiting these pathways in all of your cells and giving you all of these types of side effects. And so we are pivoting off of what others have done in the cancer field to develop our own nanoparticles with our own cocktail of microRNAs for lymphatic malformations. Excuse me. Okay, so just to summarize the things that I talked about so far. We know lymphatic malformations are characterized by this aberrant proliferation, inflammation, and this remodeling that we see in normal adjacent tissues. And our multi-omic analysis and our validation experiments identify several of these triads, these microRNA, messenger RNA, protein triads that control these processes, namely proliferation through PDCD4, microRNA 21, extracellular junction integrity through microRNA regulation of angiopoietin 2, and then also cell-to-cell adhesion through CD44, immune cell signaling, endothelial cell to immune cell signaling through ICAM1, and extracellular matrix remodeling through relin and TPA. I've shown you that inhibition of microRNA 21 in LMECs can shut off proliferation by restoring PDCD4, and that was done in vitro. And then in vivo, in an in vivo mouse model, we showed you that restoration of angiopoietin 2 expression in LMECs inhibits the formation of these dilated lymphatic vessels in a mouse model. I've also got some data that shows that if you knock down CD44, by giving it the microRNA 21, you significantly decrease proliferation as well. And if you knock down ICAM1, that experiment I showed you earlier with the adhesion of monocytes, that also is significantly inhibited. And right now we're at the process of fine-tuning like the right amount of microRNA to show us the maximum amount of this effect before we transition into the mouse model for those two proteins. All right, so what we're doing now, I've got some promising preliminary results that when you inhibit CD44 expression, like I said, you get this decrease in proliferation, and ICAM1 reduces leukocyte adhesion. We've designed these nanoparticles. We've been able to load them with the microRNAs, and we're in the process now of getting them to work in vitro. Because we want to show that you can at least get these microRNAs to be released from the nanoparticles and get into the cells. And once we have that, optimized, then we can make the transition over to mice. And we'll do that in two ways. We'll inject these nanoparticles into the Matrigel implants. But the real thing I want to do is the topical application. So we have a couple of different ways that we can do that. There's a few petroleum-based vectors that we can use, and then we'll use DMSO as well, and see which ones get across the easiest. And in terms of transfection efficiency, are we able to target the same mechanisms that we were able to do in vitro? For the extracellular matrix remodeling proteins, what I'm doing essentially is we're genotyping all of the lymphatic malformation endothelial cells that we have with ddPCR to find out which mutation they have, and then grouping them by mutation and just collecting their conditioned media. And my plan is to use that conditioned media to not only grow normal cells in it to see what happens, but also to do proteomic analysis of what is in that media and see, number one, is it different from HDLEC media? But number two, one of the focuses we have in our laboratory now is to try to figure out the differences between the cells that have the different hotspot mutations. The fact that some patients are responding to these systemic treatments and some are not, and the fact that there are three different, seemingly different, like functionally different mutations in terms of where they're located on the gene itself. That leads me to believe that the genotype, I think, matters. We just don't have enough data to say, okay, alpelisib will work for this child because they have the H1047 mutation. We're not there yet, but I think we will be when we have enough data to actually have a power to show that that's the case. There's another interesting finding that we're investigating. If you look at the breast cancer literature, there's a lot of CD44 research. If you look at breast cancers that have a mutation in PIK3CA, in those cells, they increase their expression of CD44 significantly. And those cells are highly resistant to rapamycin and alpelisib compared to when you decrease CD44 expression. So those authors were able to knock down CD44 and show that alpelisib and rapamycin were much more effective at inhibiting proliferation. So my instinct is that either CD44 is either cooperating in some way or independently is making these cells potentially resistant to our systemic treatments. So we're in the process of seeing if we can demonstrate that in vitro by comparing the activation of the PIK3CA pathway in cells with CD44 manipulation. That data is not done yet, but I feel like we're going to see something significant. At least I'm hopeful, because if that's possible, there are drugs that target CD44. They were trialed in cancer, but were abandoned due to side effects, but they were only doing CD44 inhibition. And so my question is it possible to concurrently inhibit CD44 as well as give alpelisib or rapamycin at much lower doses than we are currently giving to see if those two drugs can cooperate in terms of getting these cells to respond to the medication? And then here's my list of funding. Like I said, we have an R01 that was funded almost 2 years ago now. I can't believe that. I had a very good amount of support from the institution here when I came on. I became a project leader on a COBRE, and that enabled me to basically have protected research time in the beginning when I started out. My MD PhD student, he got an F30 from the NIH. That was the funding behind that angiopoietin 2 paper. And then we have several others that I just didn't have time to list. But for the most part, I feel like my approach to figuring out what's happening is to essentially look at other pathways that are downstream potentially of this PIK3CA activation. It's possible that in the beginning, that mutation is enough to cause the formation of these lesions, but it may just be the way you start the engine. You may activate pathways that then become constitutively active that even if you go in later with alpelisib or rapamycin or any other PIK3CA inhibitor, you may not have an effect because you're not targeting the pathway that's now aberrantly expressed. And so my goal is to develop a three-pronged microRNA treatment that targets proliferation, inflammation, and ECM remodeling concurrently with a mixture of different microRNA mimics or inhibitors that gets you the maximum amount of phenotype for the dose, and then deliver that in a novel vector that's topical. If not topical, then intralesional at least, but to avoid the administration of systemic therapy that's targeting a very important pathway that functions in normal tissues. And when you inhibit that, you get all these other side effects. And that's why we're not seeing as much adherence with the medication, and we can't go up on dose, and we don't know how long to give it. And so my hope is that at the end of a couple of decades, that we're treating these topically, but with a molecular therapy. And that's really all I've got. Thank you, guys.
Hi, Graham. Great presentation. It was very clear and really exciting work. And there's several applause coming through.
No, thank you. Where's my
and I'll just get the discussion started. Again, really beautiful work. I'm curious if you've examined the impact of either alpelisib or rapamycin on the expression of these various microRNAs, and do they have different effects on the expression of the microRNAs?
So let me see if I can get I think I need to stop sharing. You see, there we go. Hold on. Okay. Can you see me now?
Mm-hmm.
Okay. So I think the question is, did we look at what happens to microRNA expression when you give these drugs? Mm-hmm. And when we did our initial omic analysis, we did not look at treated versus untreated. We have looked at that in the context of these specific microRNAs, like microRNA 21, for example.
Hmm.
And we don't see a significant change in microRNA 21 expression when you give the cells alpelisib or rapamycin. The problem is when you do it in vitro, the cells definitely change their phenotype, right? They slow down, but in terms of each individual cell, it does not seem like that microRNA changes. I have not gone back and done another omic analysis that will show whether or not I expect that there will be a variety of microRNAs that will change. The problem with that approach is that you may end up chasing your tail because microRNAs are promiscuous, right? Each microRNA sequence can target like 20 or 30 different downstream genes. And so if you're affecting a microRNA with a drug, then how do you know which one of those targets that might or might not change is, pathologic or is potentially therapeutic? But yeah, it would be interesting to see if alpelisib shuts off, one of these microRNAs, then maybe that's the mechanism. We have not done that, but what we will do is once I can demonstrate the phenotype in the cells, in other words, once I can prove, okay, if we shut down CD44, it has an effect on the cells that I'm looking for. Then I'll go back and look at microRNA expression. My philosophy is I like to try to jump to the punchline with an experiment, because if it doesn't have the effect that you're looking for, then you might be wasting your time trying to figure out mechanistic stuff beforehand. So, but it's definitely on my list of. I think at the end of the day, we're going to end up doing another bigger omic analysis, and of course, I'm sure you're familiar with the work that I'm doing with Carrie in that big Clarum study. We're going to have a lot of more because I don't have that many samples, right? I probably have maybe 100 or so patient samples, and out of those, maybe 30 where I've isolated endothelial cells because the other ones were either before my time or just we couldn't get cells for whatever reason. Once I have enough of these cells, then I want to do a much larger omic experiment. And in that group, we're going to do all kinds of stuff, different drugs, and also different microRNA treatments. And then my goal is a combination. The idea is to find what are the right 3 microRNAs. It's going to be one of each one of those big phenotypes. One for proliferation, and I suspect that will be microRNA 21. One for extracellular matrix remodeling, and I suspect that will be one that regulates TPA, and one for this junctional integrity issue. And that may be angiopoietin or maybe one of those multitude of other junctional proteins that are obviously dysfunctional in LMECs.
Great. Thank you. Tim.
Hi, Graham.
Hey,
Great.
Tim.
Hi, how are you?
Good.
Yeah, Tim Lacrosse, Cincinnati Children's. Thanks very much for that fantastic presentation and your great work. I was curious, your approach you're kind of focusing on right now is topical administration of the nanoparticles. Do you see a potential use for those nanoparticles in lymphatic lesions that are not on the surface, that you could, let's say you've got a thoracic duct malformation. Do you think the mRNA technology might work on those?
I do. So if you look at where microRNA systemic treatments are right now, primarily they're in liver disease. And the reason is that you can give naked nanoparticles systemically, and they're all taken up by the liver. So if you've got a liver tumor, it's great, right? But for everything else, that's not going to work. So what I imagine doing is tagging or binding a protein to the surface of the nanoparticle that targets that binds to podoplanin, for example, or something that's lymphatic specific. Now, I don't think I would be able to target mutated lymphatic endothelium unless I can find a signature on the cell surface, which we're also trying to do, that can identify which of these endothelial cells have a mutation and which ones don't. Because I still don't, and maybe you know more than I do. I picture the inside of a lymphatic malformation. Are 100% of those endothelial cells, do they have a mutation? If you look at the ones floating around in the lymphatic fluid, it would suggest that they do, because all of the ones that I've isolated from a fluid sample have the mutation. But when you do the actual analysis of the endothelial cells themselves from the tissue, it's much lower than that. So until I can see an experiment where I can visualize where the mutation is, I won't have that answer. But to answer your big question is, can you target a nanoparticle to a lymphatic endothelial cell? The answer, I think, will be yes. But that technology, like when I called Thermo Fisher because they make this product called vivofectamine. I don't know if you've heard of that, but that's like it's like lipofectamine that you use in the lab, but it's supposed to target like whole tissues. And they said that they're willing to customize these and see if they can get a protein on there. Now, whether that's going to work or not, I have no idea, but that would be the goal, right? Would be able to systemically administer it. Or if you didn't have that option, we can still do sclerotherapy for those intra-abdominal lesions. And so you can imagine an interventional radiologist potentially accessing the lumen and being able to infiltrate these nanoparticles as an alternative. Because, just taking them by mouth, granted, that's a goal, but I don't know how that, I think you would digest, all the, you know what I mean? Until I can figure that out, I don't know if there's going to be a pill that goes right to your lymphatic malformation, but obviously that's the goal.
Do you think how long do you know how long those microRNAs last in terms of their inhibitory activity?
Yeah, I do. So if you look at a time course, at least in vitro, when you do a transfection, you knock down the protein expression that usually happens at about 24 hours after transfection. 12 to 24 hours, you get right away within 30 minutes to a few hours, the RNA that you're targeting goes way down. That lasts for about 3 days. And then your protein it's a similar pattern, but it's delayed. And so you start seeing it creep back up at around day 2 to day 3. And so I think it would be possible to develop a therapy where you'd be able to do like a once or twice a day type of treatment if that holds true for the actual phenotypes. I don't know if those two things are related. In other words, shutting off the protein expression, I don't know if that's going to change the growth of the lesion. It does in the lab, but I don't know if that's what's happening in the patient. Because I think that, once you trigger this immune system action, like I think a lot of that microenvironment and, maybe hypoxia and other types of factors probably keep these lesions growing that have nothing to do with the original mutation anymore. And so that's why I think you have to have multiple different targets at once, not just the pathway, but also these other phenotypes.
Yeah, great. Thanks so much, Graham.
No problem.
All right, Selene?
Hey, great talk, Graham.
Thank you.
I was wondering if you considered the canonical receptors of reelin and the TPA might be mechanistically important in your in this context, like the reelin working with the VLDL, ApoER, and the TPA through LRP1. They might be contributing to the extracellular matrix remodeling as they well do in the vascular context, and whether the mRNA affecting the signaling pathways through the receptor expression, because they do take the TPA and LRP and internalize and get rid of those ligands. Did you consider that?
Well, so this is the challenge about my approach to these lesions. What ends up happening is I now have to read about all these pathways that I have no training in, right? So like what you just
there are
said,
postdocs,
I'm like
students.
feverishly writing it down, because my approach is like, oh, I've identified this looks interesting, but I'm not an expert in TPA at all. And so, and I'll be honest, that third specific aim is still in such its infancy because we've been so busy with these nanoparticles that all I'm doing on that regard is collecting media. And so if you want to collaborate on that, I would love to have your opinion because I honestly
I'm your program officer, right?
Yeah, that's true. Oh, yeah.
Yeah.
Fair enough. But honestly,
I can find you experts to collaborate.
Because I think that that type of preliminary data I think starts to spin off into new grants, right? Because each one of these specific aims may open 3 more specific aims.
I know. Yes. So where's those?
Yeah. Okay.
New grants?
Exactly. Hey, I only have enough protected time for one R01 at a time, Selena. So you have to wait.
All right. Well, good project
I'll cut my
student.
FTE in half. We'll see.
Yeah.
Thank you for the honestly, I wrote that down. I don't know.
Yeah, I think you should, because it works in the vascular. So I bet those receptors are in the lymphatic vasculature too, and they are going to get on your way if you don't look at them.
Okay, thank you.
All
Good
right.
luck.
Doganajan?
And not the easiest, so it's fine. Great talk, Graham. I'm from UBC and I'm working on like synthetic biology and molecular biology kind of realm, and I'm interested in KLA in general. So this is like your talk is basically like what I want to do for the KLA, but includes some genome editing kind of approaches and like also LMP retargeting too. So I'm actually working with a professor from Turkey that is he's like a really an expert on detargeting from liver and then retargeting to the tissues that he wants to. So I'm actually working with Tim and then like getting some samples, not samples like cell lines, and then try to kind of find targets that we can put into in LMPs and then target them. But my question is different. So I want to ask about the samples, and you mentioned that you want to do some ddPCR on them to actually get the mutations or allele frequency, I think. Have you ever considered doing a whole genome sequencing to actually get what's going on in the cells rather than just mutation kind of perspective? You could get SNVs, other SNPs that can be extremely valuable to
Yes.
understand like drag resistance, drag like preferential drag response and other things. Yeah.
That is the overall goal of this CLARM study that we're doing in collaboration with several other institutions to do this, very deep sequencing of all of these. I have not done any deep sequencing on any of these. I bought the ddPCR machine. Thank you, Selena, for the grant. I bought that because we want to be able, as we collect these samples, I wanted to identify each hotspot for each patient so that when we have their cells in the lab, I can at least group them and see if there are differences. But the answer to your question is no. Our lab has not done that, but we have contributed our samples to this large, over 1,000 patient cohort for that type of work, and that'll happen over the next 2 years in that group. So I'm hopeful that we find something out of that, but I'm not the right person to ask.
Cool. Thank you.
And I think it'll be interesting too to see if any of the samples out of these, I think you mentioned 30 different cell lines, if any of those have the BRAF mutation.
Yeah, I also am
that'll be
curious.
exciting.
We have not looked at that. I suspect that we may find, because I think that, so the BRAF, from what I recall, I remember the Perkins paper identified BRAF. Was that in conjunction with a PIK3CA mutation? Is it independent mutation?
Yeah.
Right. So we have several samples that are negative on ddPCR for all 4 hotspots that are definitely lymphatic malformations.
Mhm.
So I know that we're going to find something. I
Yeah.
don't know what yet. But also, interestingly enough, even in the, like, I don't think that those samples, at least in our multi-omic analysis, well, in the beginning, I only had 13, so I can't say that definitively. I would have to go back and do another omic analysis to see if the ones that don't have a mutation somehow are different than the ones that do. But there's a lot of noise in this omic approach that you kind of have to filter through, and that's what's great about the multi-omic. It's like a Swiss cheese model, but in reverse, right? You get all of this information that you don't know necessarily what to do with, and then you have these multiple filtering steps. My old filtering step, like I said, was going on PubMed and picking the genes that I liked and ignoring the ones that I didn't. But that doesn't work, and so I'm hopeful that not only will we find more mutations, but that the obvious goal is to be able to take a blood sample or a tissue sample from a patient and be able to tell them, all right, you need surgery, or you need alpelisib, or you need X, Y, or Z, or if you have this treatment, your odds of recovering compared to this mutation are X. And I think that's where we will end up with, but at this stage, I don't know the answer. I bet you we find more mutations.
Mm-hmm. All right. Well, thank you once again for this great presentation. And we look forward to seeing everyone at next month's seminar. So, bye,
Thank
everyone.
you so much, everybody. I really appreciate the opportunity.